Hello viewers, welcome to Biodrunks. Let's learn some aspects of biotechnology. For your reference, I'll be providing notes, PDF link in the description. This video is also available in Hindi and Telugu. For more videos and continuous learning, please like, share and subscribe. Let's start. Today we are going to do what are the tools used for gene cloning. In the previous video, we have seen what is a recombinant molecule and how this recombinant molecule is transformed into a host cell and how this recombinant molecule is amplified. Okay, so if you remember, this was how it was. This is a recombinant molecule and this is the bacterial cell with its chromosome and how this recombinant molecule is cloned into multiple copies. So in these steps, okay, we use a lot of tools. Those are nucleases, ligases, polymerases, and DNA modifying enzymes. So let's learn one by one every topic. In this video, we will be only talking about nucleases, and in the upcoming videos, we'll be touching all the other topics. So let's talk about nucleases. When I say what is a nuclease? So nucleases, what do? They degrade the DNA molecule. And how they do it? By breaking the phosphodiester bond. Okay, so what is a phosphodiester bond? This phosphodiester bond link the nucleotides. Okay, so here we have the phosphodiester bond that links the nucleotides. So nucleases degrade the DNA molecules by breaking the link between the nucleotides. So these are of two types, exonucleases and endonucleases. Let's learn about exonucleases. What does exonucleases do? They remove the nucleotide one at a time from one end. So suppose this is our DNA strand, okay, 5 prime to 3 prime. So how does it remove? DNA is a double stranded. I'm just showing it in a single strand now. So how does it remove? It removes from one end. Okay, either it removes from the 5 prime end or it removes from the 3 prime end. Okay, so according to the position where it is removing, we have different types of exonucleases. Let's talk about one by one. Let's start with lambda exonucleases. So what is this lambda exonucleases? It removes the nucleotides from the 5 prime end. So 5 prime to 3 prime end. Okay, that's how it is removing. Then you have exonuclease 1. It removes the nucleotides in 3 prime to 5 prime direction. Okay, then you have exonuclease 3. So what is this exonuclease 3 doing? It is used for removing the nucleotides from the 3 prime end. So you have exonuclease 5. So what is this exonuclease 5 doing? It is also called as REC BCD. Okay, so it acts both as a nuclease and a helicase. It has two functions. Okay, it both acts as nuclease and a helicase. So what happens is it cuts the double-stranded DNA from both ends. Okay, if suppose this is a double-stranded DNA, 5 prime to 3 prime, 5 prime to 3 prime. So it cuts from the both ends and there hence it forms your small oligonucleotides. Okay, it leads to the formation of smaller oligonucleotides. And then you have another exonuclease, okay, that is called as exonuclease BAL31. So what does it do? It forms blunt ends. Okay, so it forms blunt ends. Cuts the DNA into shorter form in the type of blunt ends. So these are the different types of exonucleases we did. Now let's talk about endonucleases. So what does endonucleases do? It cleaves the DNA from the interior. Okay, so for example, this is a 5 prime to 3 prime. Okay, this is the DNA. So how does it cleave? It cleaves the DNA from middle. This is the way it cleaves the DNA. Okay, so let's learn more about the endonucleases. So in endonucleases, you have deoxyribonucleases. Deoxyribonucleases, that is DNA nuclease, DNAs. Okay, so you can also call it as DNAs. So what does it do? It cleaves both double-stranded DNA and single-stranded DNA. Okay, it cleaves both the double-stranded DNA and single-stranded DNA, as the enzyme endonuclease has multimeric subunits. So it cleaves both at different positions. And remember, this has magnesium as the cofactor. The magnesium is a cofactor for this. Okay. So when it has the magnesium at cofactor, it cuts at different positions. Remember, when it is having magnesium as cofactor, it cuts at different positions. And at the same time, when it has manganese as its cofactor, okay, it cuts at same position. So you need to just get one single touch that how it differs. When it is having magnesium as the cofactor, it cuts at different positions. Okay, and when it has manganese as cofactor, it cuts at the same position. Then deoxyribonuclease, also called as DNAs. Now let's do ribonuclease, that is RNAs. Okay, ribonuclease. So what does ribonuclease do? It cleaves the single-stranded RNA. Ribonuclease works at single-stranded RNA that cuts at 3' end. So 3' to 5', 3' end of pyrimidine. Okay, let me write it properly. 3' end of pyrimidine. This is very important. Cuts at the 3' end of the pyrimidine. Okay, so I think till here it is very easy. Okay, now we did DNAs and RNAs, which act as the endonucleases. Now you have S1 nuclease. Okay, so what does this S1 nuclease do? It degrades RNA or single-stranded DNA. It can only degrade single strands. Okay, whether it is a RNA or DNA, it can only degrade single strand, not double strands. Okay, it's very easy. It can only degrade single strands and not double strands. Okay, so double strands means, and it also has one more thing. Okay, it can not degrade RNA DNA duplex. When I call about double strand, that means RNA DNA hybrid 2, which works during the, where it is working, it works during the reverse transcriptase, the process of reverse transcriptase, that is cDNA synthesis. The very important, most important endonucleases is restriction enzymes, restriction endonuclease. So this restriction endonucleases are the most important. Okay, so restriction endonuclease is totally a whole new topic and it cannot be dealt with some other topics. So I thought it would be better if we are doing it in a very separate video. So in the upcoming videos, once we complete the tools in genetic cloning or genetic engineering, then we will be having a particular video about restriction endonucleases, where we will be learning the type of restriction endonucleases 1, 2, 3, and 4. okay, in very detail, and what are blunt ends, what are sticky ends. So we are going to learn everything in detail in the upcoming videos about restriction endonuclease. Right now, it's very important to know that it is used as a tool for cutting DNA, okay, during RDTs. So it's a very important tool in recombinant DNA technology. So how it works? It works as it recognizes the sites that are called as recognition sites. Okay, so it detects recognition sites and it cuts the DNA according to them. Okay, it's very, very, very important. Okay, and we will be doing it for sure in a very lengthy and detailed manner in the upcoming videos. So in the next video, we will be dealing about DNA ligase. This was all about today's video. Hope you liked it. For more such interesting video, please like, share and subscribe to Biodrunk. Let's meet in the next video. Bye-bye.